Hello friends, today we are going to start with the topic genetic variance. Before starting the topic, we came across the term many times like what is variance, variation, what is these terms and what is the actual difference between them. So the actual difference means between variance and variation is variance is absolute term. Absolute terms means the terms that is used to measure something, that is variance. In statistics, it is used to measure different components like additive variance, genetic variance, and dominance variance, and epistatic variance. But what is the variation? That is this variation means how one thing is varying, how the if you are measuring the two things, so how like one is expected value, that is suppose expected expected value is something like 20, and mm, another is observed value comes like 15. So how these two values are differing from each other, how they are, what is the difference between these two values, that you call variation there. And the each value 20 or 15, we can say they, they may be a measure of variance, means if we are measuring them, they are variance. So for measuring any absolute value, we are using variance. And what is the difference between our observed value and expected value is known as variation. Okay, now first we will go with variation. So genetic variation, they are mainly of three types. That is, we can in genetic variation, in short we can write gv. Gv is mainly of three type genetic variation, that is phenotypic, sorry, we will write gv, that is mainly phenotypic, genotypic, and environmental. These are the three type of genetic variation. They are not type of variance, they are type of variation. In phenotypic variation, the phenotypic variation is include the sum of genetic variance and environmental variance. Both the variation will be included in phenotypic. Means phenotype means what we are able to observe from outside on the basis of morphology. So how the morphologically the things are varying, are differing from each other, that may be due to two reasons. Either maybe a genotype, difference in the genotype of two things that you are comparing, or they may be the effect of environment. Either the two things that you are comparing may be living in different environment, and environment effect are coming, that's why they are difference. So the phenotypic variation is mainly of two, due to two reasons, genotype and environment. Now the another term that we come across is heritable variation and non-heritable. Non-heritable, the heritable variation are those which can pass from one generation to another generation, which can pass from one generation to another generation. Suppose heritable, first we see heritable. In heritable variation, they are able to pass from one generation to another generation. They include genotypic variance. Genotypic, all the genotypic variation are able to pass. Genotype is passing from parents to offspring. So this is heritable portion of variation. Another portion of variation is non-heritable. Non-heritable variation, non-heritable, non-heritable. Non-heritable does not pass from one generation to another generation. That is mainly through environmental variation that we are seeing. So it is non-heritable portion of variation. So these are three type of variation. Now we will see the classification of genetic variance. So many people are confused between these two terms, genetic variation classification and genetic variance classification. So while answering the question, you should be carefully see what they are asking, whether they are asking the genetic variance or genetic variation. Now we will go for genetic variance. Various classification was given for um, genetic variance. First we will see what is genetic variance. The genetic variance is the inherited variation which remain unchanged by environment factor. Means this genetic variation remain unchanged by environmental factor. Means they will pass from, this is heritable portion, means they will pass from one generation to another generation, and this is the most valuable for plant breeder for exploitation by selection, because if you select for this type of variation, they will be inherited or passed to the progeny, further generation, they will be passed to further generation into the progeny. So these genetic variation, they are consisting of three types, that is additive, dominance and environment. These are genotypic variation. Now we have to see what is variance. So genetic variance, short we are writing GV, that is of three people have given their classification. First is R.S. Fisher. According to R.A. Fisher, 1918, he divided genetic variance into three components. This is additive, dominance and epistasis. These are the three components on which he divided. Now there is another scientist, that is Wright, in 1935. He divided the genetic variance again into the two fraction, that is mainly additive variance and non-additive variance. In non-additive variance, according to Wright, non-additive variance is further classified into dominance and epistasis. So these are the three type of variance classification. Now again one new scientist came and he classified the variance in two components, that is heritable, 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 fixable, in which he include additive variance and additive into additive type. Another is heritable non-fixable, which is dominant variance, additive into dominance and dominance into dominance type epistasis. These are terms I will describe one by one clearly. Now first we will go with heritable. Heritable, as I already described, heritable is what can pass from one generation to another generation. Fixable. Fixable means which we can fix. Means if it has if fix means like the intensity of that component will be fixed. Suppose your any character you can suppose in plants like any genetic resistance, like you can suppose drought resistance, drought resistance that may be due to any morphological character you have selected, like for drought plant is resistance in mainly we can say like leaf, you have seen like leaf rolling is the character. Now if you are seeing the leaf rolling as a character and it is passing from one generation to another generation, that is heritable. But while passing from one generation to another generation, is it that amount of leaf rolling is same? मतलब जितना लीफ रोलिंग हो रहा है मीन्स like suppose in drought half leaf is rolled, sometime all the leaf means all the leaf will get rolled, some means their amount or intensity will be vary according to that they are fixable, means they are able to fix or not. So according to that he given two component, heritable fixable, that is additive variation, and additive into additive type epistatus. Means these are the two components which are heritable also and fixable also. Then he divide heritable non-fixable. In heritable non-fixable he has divide dominance variance, and additive into dominance and dominance into dominant type epistatus. So these are the mainly three type of classification that is given for genetic variance. Now each terms we will discuss one by one. First is additive variance. What is additive variance? Additive variance is the component of genetic variance that we have already seen. Genetic variance is only classified into three: additive, dominance, and epistasis. So what is mainly the additive variance means? It is the average effect of gene on a all segregating loci. Means on a segregating loci, what is the additive average effect of gene? Suppose you are having two loci, A, capital A, capital A. So what is the average effect of these two loci? That will give you the average effect of each loci will give you the additive variance. And additive variance is mainly due to, suppose you are having two loci, capital A, another is small a, small a. These have different average effect, these will have different additive difference effect. So what is the difference in this effect of two homozygotes? Both are homozygotes. So what is the difference between these two homozygotes? This will give the measure of additive variance. So additive variance expression, they show lack of dominance, means intermediate expression they will show. In natural plant breeding population, additive genetic variance is predominant, one closely followed by dominant variance. Means additive variance in natural population, which variance is more? Question can be asked. So additive variance is more in natural population as compared to all the three variants. Additive, what are three type? Additive, dominance and epistasis. Among all type of variants, additive variance is more. Additive variance is associated with homozygosity and therefore it is expected to be maximum in self-pollinated species and minimum in cross-pollinated. So maximum in self-pollinated. Additive variance is maximum in self-pollinated species and minimum in cross-pollinated species. How we will measure this additive variance? So additive variance is measured by various biometrical techniques. These are, the component is mainly GCA. Additive variance is measured by GCA. GCA is what? Nothing but general combining ability. This will describe in further in our upcoming chapters what is GCA. Now, effect on selection. Selection is very effective with additive genetic variance because it is fixable. As previously I have told, the Mathur has given the classification heritable and fixable, in which additive variance is included. So it is fixable. So the things which are heritable and fixable also, so they are very good for selection, because you will select them in parent population, they will be definitely will be transferring to offspring also, and they will fix in offspring also. That's why it is the main component of selection, and it's mainly exploited during selection use. Additive genetic variance is measure of breeding value of a genotype, and it is used for estimation of heritability in narrow sense. Heritability mainly measure in two type, broad sense and narrow sense. So narrow sense heritability is measured by additive genetic variance. That this type of heritability also we will see in our upcoming class. Now transcription segregation result due to additive genetic action when the parents are intermediate to extreme value of the segregating population. So like you have seen, additive transcription segregation is also due to, like if you are having 2A type of homozygous progeny, next upcoming generation what they will give, capital A, when you will, and small a, when you will further self them this capital A small a, what type of progeny you are going to get? Capital A, capital A small a, and small a. So these are mainly the component of transcription segregation due to additive genetic variance. Now the second component is dominance variance.